The four colors, green, red, blue, and black, each represent one of the four nucleotides, A, T, C, and G, respectively, and the sequence is printed on the top of the plot. In this figure, we start reading our DNA sequence from position 28 base pairs after the CGCCCTT site, which is usually recognized as a starting point for targeting DNA sequence reading. As for this clone we made, our expected DNA sequence should start from GAA, TCC, AT, GG, AC, ACC. So we need to check each nucleotide one by one in order from position 28 on the figure, such as the G at position 28, which should match our expected first nucleotide as G. Yes, they match each other, but sometimes the DNA sequencer may make some errors. For example, the second and third nucleotides we expect should be two A's, but we can only find one at position 29 on the top of the plot. So is there another A missing? In this case, we need to check those nearby nucleotides to see if there is any overlap reading, which may read two nucleotides as one, and thus miss another one. To check with this, we first need to adjust the sequence plot so that we can examine it more clearly, and then look at the nearby nucleotides such as position 28 and 30. Yes, there is an overlap reading of G and A at position 28. By increasing both the width and height of the plot, we can easily observe a relatively low peak in green, which represents A, under a high black peak representing G at position 28. Then we make sure that the first three nucleotides sequenced are all correct as expected, GAA, and then we can move on to check with the next.